This video lecture will cover Chapter 6, which is about proteins and amino acids. Again, there will be some slides that have been omitted that you may see on Moodle in the PowerPoint, so don't fret over anything that may not be included. So to begin, what exactly are proteins? Basically, these are substances that contain carbon, oxygen, hydrogen, sulfur, and specifically nitrogen. So nitrogen is the key component here in these proteins. We all know proteins are composed of amino acids, and basically this translation of amino acids helps to build specific proteins. We talked about this at the beginning of the semester. So we have our essential amino acids and our non-essential amino acids. In our fatty acids chapter, we talked about our essential fatty acids versus our non-essential fatty acids. Our essential fatty acids, remember, were those that we couldn't create in the body, right? We didn't have that enzyme to code past that 9th carbon. So, for instance, that's an example. Our essential amino acids are similar in that we can't create those in our body, right? So they're essential to our diet. We have to consume them. So here you see the list of our essential amino acids. And then others are non-essential amino acids. It is important to note that there are hundreds, or I should say like about 140 amino acids that exist in nature, but only about 20 are significant to the human body. That just means that 20 of those are actually utilized in the human body and important to our physiology. So I mentioned essential amino acids. These are the amino acids that are required in our diet because we can't synthesize them specifically in adequate amounts for growth and maintenance. So some we may be able to synthesize in small portions, but not enough to meet requirements. So, for instance, some of these essential amino acids can be present in foods that we consume, but not in sufficient amounts to support that growth or maintenance. So, for instance, there could be a protein food that we consume, but it may be limited by the specific essential amino acid that isn't in a large enough quantity. And in that circumstance, we would call that a limiting amino acid. So, I'm sure you've heard before, proteins can come from animals or from plants. Of course, our plant origin proteins typically are going to be lower in protein and lower in biological value, whereas our animal proteins have usually a greater protein content, period, but they're also more biologically available to us. And that is because of how much more difficult it is to break down plant cell walls and release that protein during digestion. So speaking of digestion, right, we have to be able to break down these long, big, complex proteins into smaller, more digestible and absorbable materials, right? So if you think about our long polysaccharide chains, we had to break those down to eventually form monosaccharides and disaccharides. So similar with our proteins, we want to digest those down into smaller peptides or individual amino acids. So with that, we're going to use digestive enzymes specifically made for proteins. In this chart that you'll find in your book, you can see specific enzymes that are important in protein digestion, where they work in the body, and then you'll see endo or exopeptidase here. Basically, this means this is where that enzyme is working along the peptide chain. So endopeptidase means it's working on the inside of the chain, whereas exopeptidase means it's working on the outside of the chain. And you'll also see here that there are specific amino acids where that hydrolysis will take place from that enzyme. So our enzymes are looking at specific amino acids to work on. It's not just random. So like I alluded to earlier, amino acids are going to be the primary way that we absorb proteins. We can, however, absorb them as dipeptides and oligopeptides as well, and even some intact protein. This is more common or more likely in infants. If you think about that, you've always heard that the breast milk, or specifically colostrum, is important to infants to transfer mom's antibodies, which are proteins, to baby because baby doesn't have that immune system. So this right here is an example of how humans can absorb whole intact proteins rather than just amino acids. Now, as we age, we won't be able to absorb those whole proteins anymore as our digestive system matures, but this is an important time during the life cycle that we are able to absorb those intact proteins. So, also similar to carbohydrate transport, amino acid transport is going to use this concentration gradient with a carrier-mediated mechanism. So we're going to have an amino acid carrier along with sodium and then vitamin B6 to transport the amino acid from the lumen into the enterocyte and then out of the enterocyte into the blood to be transported throughout the body. ATP is required in this reaction as well. Here this shows you a graphic of what I just mentioned. We have our amino acid here with our amino acid transporter and sodium carrying in the amino acid from the lumen into the enterocyte. You'll see this sodium-potassium pump is also working along with ATP to maintain the a higher sodium concentration outside of the cell. And then we have, to get our amino acid from the enterocyte and into the blood, we also have this amino acid transporter and sodium working as well. So I already mentioned our high quality proteins and our low quality proteins. Basically, this is the amount of essential amino acids that are contained in that food product. So to assess the quality of proteins, oftentimes we'll use reference proteins, and those can be egg or casein typically. Those are our usual reference proteins. We also will assess the protein quality by calculating either the biological value or the net protein utilization. Both of these are going to be an estimate of how much protein we're using in the body and how efficiently we're using it. So biological value is basically looking at how much protein we consumed, how much is digested, how much is absorbed, how much is actually going to be used in the body versus how much is not. Net protein utilization is looking more into how efficiently that protein is utilized. So you'll have to look at this in your book, the different formulas for these, and you'll be able to decipher between them as well. So I did mention earlier some enzymes, or how enzymes work on our proteins to help with metabolism. Just look at these and be able to recognize them. Just like any other macronutrient in our body, protein has a lot of important roles, not just in our muscle building, but these can range from, blood clotting factors like thrombin and prothrombin, other plasma proteins, and hematologic components. They help to, of course, with our muscle contraction, but they also help to form hormones, connective tissue. They're very important in our water balance, and I'll talk about that in a second. Also with maintaining our acid-base balance, building antibodies, which are proteins again. And then if you think of everything that is a protein in our body, from enzymes to transport carriers to hormone receptors, right? All of these are proteins, so all of these are important roles and functions of proteins in the body. So there are some amino acids, or ways that we can classify amino acids. They can be either ketogenic or glucogenic. Basically, this means what that amino acid is able to do once it's metabolized. So we have this, if you think of the glycerol backbone, so amino acids have this backbone as well, this carbon structure. So after the amine group is removed, that carbon structure can go either into the Krebs cycle to eventually become glucose, or it can be utilized to form ketone bodies. So those amino acids that are able to be used to form ketone bodies are called ketogenic amino acids, and those used to be able to form eventually glucose are glucogenic amino acids. There are a few that are able to be both ketogenic and glucogenic, and again, you can see that in your book. Another important role of amino acids is that they can be precursors for other biochemical compounds. So that means they can form other amino acids, they can form hormones, they can form neurotransmitters and bile. Let's see. One important function is to be able to donate carbons in transfer mechanisms. Methionine is particularly important in donating those carbons. And you'll see SAM here is the principal methyl donor. So if you think about a methyl, that's a carbon with those hydrogens attached to it, right? It's a little unit, and it's used in lots of different substances or compounds in the body. So methionine is within this SAM that's donating these methyl groups. And that's to help create those molecules that we mentioned in the previous slide. So to be able to actually use amino acids as energy, we have to go through these degradation mechanisms. So that can be transamination and deamination. Basically, with transamination, we're transferring that amine group from one spot of the amino acid to another. And then with deamination, we're removing that amine group so that it can be utilized as energy. So this is just showing you transamination and deamination. So with that, we want to be able to synthesize amino acids that are non-essential to the diet, right? So glutamate is going to provide most of the nitrogen for those non-essential amino acids. If you look at the Krebs cycle, you'll see that a lot of amino acids come out from those from the Krebs cycle as intermediates. So like I mentioned before, if we take off the nitrogen from amino acids, we have this carbon skeleton. Once carbon skeletons are degraded, they can be entered into the Krebs cycle or into eventually becoming ketones, and that's what we talked about, the ketogenic or the glucogenic amino acids. And you'll see those lists here. They're glucogenic, are exclusively ketogenic, and then are combined ketogenic and glucogenic amino acids. So glutamine is another notable essential amino acid in that it can transport nitrogen and carbons between many organs. Glutamine is found in most tissues, typically built in the skeletal muscle, lungs, and adipose tissue. We'll talk about ammonia with their urea cycle, but this is actually, glutamine is actually one way that we can dispose of some ammonia. And of course, ammonia is the byproduct of breaking down our amino acids. We also can use glutamine by the intestinal cells, either as energy, but it can also be formed like a protective barrier. If you look at information on like leaky gut syndrome and things like that, glutamine is always kind of popping up, and that's why. Not 100% valid. I mean, leaky gut syndrome, but it's always going to pop up because it is an important part of it. Okay, so one thing we mentioned about amino acids was their ability to form neurotransmitters. Specifically, tyrosine is able to form our catecholamines, which is our DOPA, dopamine, epinephrine, and norepinephrine. And these catecholamines, what they do is they're excitatory, so they help to increase the metabolic rate. And you'll see people treated with medications that help to decrease degradation of tyrosine, so that you're able to utilize tyrosine to make more catecholamines to help prevent things like anxiety and depression. Other notable one is tryptophan, which can be converted into melatonin and serotonin, which have other factors with mood and sleep, of course,